Okay, hi. This is our lecture on cell signaling, which is chapter 17 in the new version of the textbook. Okay, so in terms of, well, just a little bit of introduction, right? In the last lecture, which was on plasma membrane, right, we talked about, or I talked about ion channels, right? And there was one example where I showed you a neurotransmitter being released by one neuron and then it going into the synaptic cleft and then binding to receptors on the other side, which I said, imagine that was a muscle cell, right? And then we could think of that neurotransmitter as binding to a receptor, and that receptor is basically an ion channel, right? And so that was a ligand gated ion channel. When that neurotransmitter binds, it would cause opening of that ion channel, okay? So now imagine a neurotransmitter binding, except that when it binds to its receptor, it's not just going to be an ion channel, it's going to initiate some other different type of response in that cell. Okay, so that's what this chapter is talking about. It's talking about cell signaling. So we'll talk about different types of signaling molecules, neurotransmitters being one of them, but there are others as well, and their receptors. And then different examples of what might happen when a ligand binds to a receptor. So it's dependent on what type of molecule it is. Is it a neurotransmitter? Is it a hormone? It's also dependent on what type of receptor it binds to. Okay, so now we're sort of talking about what's happening in that cell as a response, right? So there's some molecule, think about, if you want to think about a hormone traveling in circulation, it knows its target cell based on its ability to bind to its specific receptor on that target cell. And then what is happening in that target cell. Okay, so that's the little bit of intro and kind of tie in from what we talked about last. So here's a breakdown of what we're going to cover. And remember, I did cut out quite a bit from this chapter. And remember that there is a lot of information regarding cell signaling in general. Okay, and so we're just getting a little snippet of it. Okay, so we'll first start talking about signaling molecules, different types of signaling molecules, like I said, and the receptors. We'll particularly talk about G proteins and G protein coupled receptors and how what signaling occurs from those receptors. Also, we'll talk about tyrosine kinases and other kinases as well, okay, and some other pathways. Okay, so it says all cells receive and respond to signals from their environment. So I just gave you an example of think about a hormone traveling through the bloodstream. It needs to be able to bind to its target cell, and then so that's the target cell receiving that signal, and then there's some sort of response that would be initiated. Okay, it says bacteria and unicellular eukaryotes will respond to environmental signals, right? That are basically, coming from outside of that actual bacteria, right? Or the actual organism. And those will then bind to receptors that are sitting on their surface, okay? And so it says signaling molecules that might be secreted by other cells, right, for mating, but then also for other communication as well, okay? Obviously, we're going to be focusing on multicellular organisms and focusing on cell-to-cell communication, okay? It says each cell must be carefully regulated to meet the needs of the whole organism, right? Think about that. Think about how different systems in your body function, right? Remember, when we think about physiology in general, you have to think about what's happening on that cellular level, right? And so what's happening in each cell has to be carefully coordinated so that then that whole organ, right, or that whole system can work, or that even that whole tissue can work. Okay, so it says a variety of signaling molecules are secreted or expressed on the surface of one cell and bind to receptors expressed on other cells. Okay, so bottom line is there's not just one single, molecule that's released that then just binds to one single receptor. There are multiple if you're talking about any one given cell type. Okay. All right, so it says binding of signal molecules to the receptors initiates a series of reactions that regulate all aspects of cell behavior. So this is what I was sort of talking about in the intro. So whenever that signal molecule, whatever molecule it may be, a neurotransmitter or a hormone, binds to their receptor, right, there's going to be a series of reactions that are initiated inside that target cell, okay? It says many cancers arise from problems in signaling pathways that will control normal cell proliferation. Okay, makes sense, right? So if there's an issue in some signaling pathway that's involved in controlling normal cell proliferation, and there's a problem with that pathway, then maybe there is proliferation that becomes out of control. Okay, and when we our next chapter on cell cycle, we're going to talk about some of these things as well. Okay. All right, so it says much of our understanding of cell signaling has actually come from cancer cells. Okay. Okay. So in terms of different types of signaling molecules, okay, so this is the first part of this lecture. Basically, what are different types of signaling molecules? I've already mentioned some. Okay, so they can range from simple gases, okay, to really complex proteins. Okay, so some can carry signals over long distances, others just locally. So when I gave you that neurotransmitter example, that is an example of a molecule that just acts locally. That neurotransmitter is not released into the bloodstream and does not travel long distances. Okay, these signaling molecules can also differ in their modes of action, right? Some can cross the plasma membrane on their own and actually bind to intracellular receptors. Others will bind to receptors on the cell surface. So again, there's lots of variety, okay, depending on what molecule you're talking about, and again, what target cell, what receptor type, and what have you, okay? All right, so modes of cell signaling: direct cell-to-cell signaling, okay? So these are just different ways that cells can signal, okay? So it says direct interaction of a cell with its neighbor, okay? So via things called integrins and cadherins, okay? Signaling by secreted molecules, okay? And there's different categories of secreted molecules that we're going to talk about in a second, which are based on how far they travel, essentially, okay? All right, so if we talk about direct cell-to-cell signaling, this is, you can think of, so it's not a secreted molecule. Think about, I guess you can kind of think about this green part here as being a molecule that's bound to the surface of one cell that then binds to the receptor on another cell, okay? So this is cell-to-cell signaling, okay? So this is going to initiate some sort of intracellular reaction or initiate an intracellular cascade in this cell once it binds, okay? But again, the difference is that this is not a secreted molecule. This signal is bound. The signal's bound, and so is the receptor, okay? All right. If we talk about signaling by secreted molecules, here are some examples, okay? So endocrine signaling involves hormones, okay? These are secreted by endocrine cells, right, that are in endocrine glands, and they actually get released into the bloodstream, right? So they're carried through circulation, and they then can act on target cells at very distant sites, okay? So for example, estrogen. And actually, I mean, estrogen, can be produced and released by the ovaries, right? But there's also estrogen receptors in the brain, okay? So once it's released into the bloodstream, it can then go and bind to its target cell wherever that may be, or target cells, right? Depending on where those are. Okay, so again, endocrine signaling, these are hormones that are released by specialized cells that are in endocrine glands. They then get released into the bloodstream and they're carried in the blood to a distant target cell. Okay, so once it meets its target cell that has its appropriate receptor, that molecule will then bind and initiate some response in this cell. If we talk about paracrine, so that was endocrine signaling, right? Hormones released into the bloodstream travel really far distances. Paracrine, okay, these are molecules that are released by one cell and they only can act on neighboring target cells. So the example for this is neurotransmitters, right? So again, some cell releases a signal, it then can just go and bind to neighboring cells because basically it's just by diffusion, right? It can only diffuse across short distances. Eventually it will be degraded. And so it's only able, like I said, to go short distances. Okay. These do not enter into the bloodstream and they don't have distant targets. So again, the best example of paracrine signaling is neurotransmitters. Okay. Or are neurotransmitters. All right. Autocrine signaling is another example. Okay, so this says cells respond to signaling molecules that they themselves produce. Now remember, these are all examples of secreted, right? Signaling by secreted molecules. Okay, so in this case, this is something that's secreted by a cell that then has a response on itself. Okay, so example here, T lymphocytes respond to antigens by making a growth factor that drives their own proliferation, thereby amplifying the immune response. Okay, so when they encounter an antigen that stimulates them to produce a growth factor that they released, that growth factor can then go back and bind to those T lymphocytes and induce proliferation of the T lymphocytes. Okay, again, in terms of the tie to cancer here, there's a lot of abnormal autocrine signaling that will contribute to cancer. Again, involving growth factors. Think about it, right? What do growth factors do? They stimulate proliferation. So it makes sense that if we're going to talk about cancer, a lot of the or where there would be a problem in essentially in cancer, it would be with some sort of pathway, right, that's involved in increasing proliferation. And a lot of these involve growth factors. Okay, so it says a cancer cell can produce a growth factor, which will also, so it's secreted, right, secretes it. That growth factor can then bind to that same cancer cell and stimulate its own proliferation, okay, and also stimulate other cells around it to proliferate. So pretty smart on the cancer cell's part. Okay, so that's what this is showing you. So here's the same cell releasing a factor that then goes and binds to itself. And again, if you want to think of an example related to autocrine signaling, think about regulation of proliferation. Okay, and so on the good side, T lymphocytes would depend on something like this to be able to amplify the amount of T lymphocytes that are produced, right, to respond to a particular antigen. However, cancer cells sort of use the same mechanism to basically increase their proliferation. Okay, so that's not such a good application. Okay, now if we talk a little bit more now about the receptors and different types of receptors, okay? So those were we all just gave examples of different types of signaling, okay? Right, and different types of secreted signals. All right, we also talked about direct cell-to-cell signaling, okay? All right, now we're going to talk about receptors, different types of receptors. Okay, so receptors can be located on the cell surface, which is probably what we most often think of, but they also can be located inside the cell. And I know I've sort of given you guys that example before and sort of told you that there are intracellular receptors. Now we're going to talk a little bit more about that, okay? Intracellular receptors will respond to small hydrophobic molecules that can diffuse across the plasma membrane on their own. Good example, and the example I've given before, probably in Bio 111 and also in here, are steroid hormones. Okay, thyroid hormone also can diffuse across that plasma membrane and has an intracellular receptor. Vitamin D3, and another example is retinoic acid. Okay, so these are examples of molecules that can diffuse across the membrane on their own. Therefore, there's no need for the receptors to be sitting on the cell surface. Okay, all right. So it says receptors for these molecules, so molecules that can cross the plasma membrane on their own, okay, their receptors are members of a superfamily called nuclear receptors, okay? They are themselves transcription factors. So take a pause, think for a second, what's a transcription factor, right? A transcription factor is a protein that can bind to DNA, right? And then it will change transcription levels. It might activate, right, transcription, or it could inhibit transcription, right? So they have domains for ligand binding, and then DNA binding, right? So think about it. It's a receptor sitting inside the cell that the ligand will bind to activate. Once that ligand binds to that intracellular receptor, that sort of complex can go to the DNA and bind to the DNA and activate transcription, okay? So steroid hormones and related molecules directly regulate gene expression, right? Think about that. They're directly regulating gene expression because when that steroid hormone binds to the receptor, that's a transcription factor that then goes and affects gene expression, right? Affects transcription. Okay. Now, there's obviously, it's not just one size fits all with any of these situations or any of these examples, by the way. And again, remember, I'm telling you, I'm only just giving you a few examples of these. There are so many different varieties of these that you can just spend hours and hours going over these different pathways and these different types of examples, okay? So it says at the top there in bold, ligand binding has different effects on different receptors, okay? So again, the response that's going to happen inside that target cell is dependent on what type of receptor the ligand binds to. It's also dependent on what type of ligand binds to the receptor, okay? There are lots of different varieties and different types. Okay, so it says here, for example here, some nuclear receptors are inactive in the absence of a hormone. Okay, so if the hormone isn't bound, then the nuclear receptor is inactive. Okay, so it says glucocorticoid receptor is bound to heat shock protein 90 chaperones in the absence of a hormone. So there are these things that are called, they're basically called binding hormones. Okay, so anyway, think about the glucocorticoid receptor is inside the cell, but it's bound with these other proteins, these heat shock proteins. Okay. When a glucocorticoid will enter, right, the steroid hormone enters into the cell, right, because it diffuses across the plasma membrane, it will displace this heat shock protein 90 protein, or this binding protein, and then it will activate the receptor and allow that to act as a transcription factor and bind to the DNA, okay? So here's an example. All right, of this. So this is showing you glucocorticoid action. So glucocorticoids, right, like cortisol. Okay. Again, this is sort of a generalization of how some of these work. Again, there are many sort of different alterations of these. Okay, so here's your glucocorticoid, right? It can cross that plasma membrane on its own, right? It diffuses across the membrane. Okay. And it will then, so here's your glucocorticoid. It can diffuse across the membrane, all right? Before that, or if that glucocorticoid is not present, right? Again, like think of cortisol, the stress hormone. This glucocorticoid receptor sits here inactive, right? Because it has this heat shock 90 protein bound to it. So in this state, the receptor is inactive. However, if glucocorticoid enters into the cell, it will basically displace this heat shock 90 protein and bind to the receptor. It is only active when it's bound by the glucocorticoid. You also notice there's sort of another step, which I didn't really mention, but you can see from this diagram that it then dimerizes or forms a dimer. So we've talked about dimers before, but basically it's two of these active glucocorticoid receptors coming together, okay? Now, what do you notice happens next? This then binds or enters into the nucleus, right? And binds to the DNA and acts as a transcription factor. If you also notice here, this has a co-activator, HAT, right? What's that? Think about that. Histone acetyltransferase, right? And so what does histone acetyltransferase do? Adds acetyl groups, right, to the histones, which helps open it up and increase transcription. Okay, so you can see how this then is going to enhance or increase transcription of specific genes. Okay, so do we need to memorize any of these? I mean, this is the first one we're talking about. No. Again, I mentioned this too during our Zoom meeting, but you know, a lot of the essays that are pertaining to this chapter, there will be pictures like this where I ask you questions about the picture, and those questions are getting at if you're understanding what this is showing and what's happening here. It is not about memorizing. Please do not memorize any of these pathways, okay? But understand. Okay. All right, so here's another example of a molecule that can cross the plasma membrane on its own, okay? And maybe something that maybe you never really heard about, but it's nitric oxide, right, or NO. And it's a paracrine signaling molecule in the nervous system, immune system, and circulatory system. So when we say it's paracrine, what does that mean? It means it only will act on local cells, okay? But it can actually cross that plasma membrane and bind intracellular, okay? It says here it'll alter the activity of enzymes. NO is synthesized from arginine, Okay, again, its action is local because it is extremely unstable. So it's actually even more unstable than other types of paracrine signals. Okay, so it says its half-life is only a few seconds. So as soon as it diffuses across, or as soon as it gets into the outside of the cell or in that intracellular space, or sorry, extracellular space, it would have to really bind immediately to another cell or adjacent cell that's there. Otherwise, it's going to be degraded very rapidly. Okay. All right. So in terms of the target of NO, okay, its target is guanylyl cyclase. Okay. So again, we said it's basically going to alter the activity of enzymes. Okay. So one of its targets, or main target, right, is guanylyl cyclase. Okay. So when NO binds, it will stimulate the synthesis of a second messenger. And we're going to talk about second messenger again, and we should be able to pick out what the second messenger is from some of these pathways based on diagrams. Okay. So it stimulates the synthesis of a second messenger called cyclic GMP. You'll also see that we'll have another second messenger that we'll talk about, which is cyclic AMP, similar molecule. Okay. So in terms of what a second messenger is, again, we should understand what a second messenger is. It is a molecule that relays a signal from a receptor to a target inside the cell. Okay. So just as the name implies, it's a second messenger. NO, so in terms of some of the effects of NO, it can signal dilation of blood vessels. Okay. So neurotransmitters act on endothelial cells to stimulate NO synthesis. And then once NO is synthesized, it would cause dilation of the blood vessels. Okay. NO diffuses to smooth muscle cells and stimulates cyclic GMP production. That then induces muscle cell relaxation and blood vessel dilation. Okay, and so this is basically the mechanism in terms of what it does, right? So we said it can signal for dilation of or cause dilation of blood vessels. How does it do that? It basically acts as the second messenger, right? And stimulates the synthesis of the cyclic GMP. And that's what then induces that muscle cell relaxation and blood vessel dilation. Okay. So I'm not showing you a picture of it in terms of the specific pathway, but I guess I just want you guys to sort of be aware of the effect of NO. Also understand that nitric oxide, NO, is a paracrine signaling molecule. Okay. It can cross the plasma membrane on its own, and it will then, one of its main targets is this guanylyl cyclase, okay, which then stimulates the synthesis of the second messenger. What is an effect of NO? What is one of the effects that it has? Blood vessel dilation, right? Smooth muscle cell relaxation and blood vessel dilation, right? You can kind of think about that. That's together, right? If we think about blood vessels, right? We have that lining of smooth muscle. And so if that smooth muscle relaxes, that's going to cause those blood vessels to dilate. So NO essentially has an effect on smooth muscle cells, okay? And then that then causes those blood vessels to dilate. All right. Now, let's talk a little bit more about neurotransmitters. Again, these are paracrine signals, okay? They carry signals between neurons, right? Or from neurons to other cells. Again, if you think back to my example that I started this chapter with, my example that I gave during when we were talking about the action potential and ligand-gated ion channels, it was the neuron, right? The neuromuscular junction, the neuron stimulating a muscle cell on the other side, right? So you can think of neurons can stimulate glands, they can stimulate muscle cells, right? And they also, or not stimulate, regulate, or because it's not always stimulating, right? But basically they signal. So right, so a neuron can send a signal to a muscle cell. A neuron can send a signal, and it's in the form of a neurotransmitter, to a gland, right? A cell in a gland. A neuron can send a signal to another neuron. Okay. All right. So it says the release when an action potential arrives at the end of the neuron. I mentioned that And again, we didn't get into crazy detail about this. This is something that I obviously focus on a lot in neurobiology, okay? Once that action potential travels down that axon and reaches the end, it causes neurotransmitters to be released, okay? Those neurotransmitters then diffuse across the synaptic cleft and they bind to receptors on the target cell surface, okay? Here are some examples and structures of neurotransmitters. Nobody needs to memorize structures or anything like that. They're just there to sort of get an idea of what some of these chemical structures are. The one example that I had given you guys again at the neuromuscular junction was the neurotransmitter acetylcholine. This was actually the first neurotransmitter that was discovered. These are some other examples of neurotransmitters. So we should be familiar with at least one other example of a neurotransmitter. Maybe two other examples. Glycine, glutamate, dopamine, norepinephrine. Many have heard of some of these before. Serotonin, epinephrine, histamine, okay, GABA. These are just some examples of neurotransmitters. Okay, it says neurotransmitters are hydrophilic. They can't cross the plasma membranes on their own, okay? So they bind to cell surface receptors in general. I will say that you will see that NO can be considered a neurotransmitter in some sense, or some people, or you'll see it as listed as a neurotransmitter because we said it's a signaling molecule in the nervous system. So just so you kind of know, that's sort of a side note, I guess, that you could consider NO as a neurotransmitter, okay? But in general, most neurotransmitters have to bind to a receptor sitting in the plasma membrane, okay? Says in the second bullet point there, many neurotransmitter receptors are ligand-gated ion channels. I gave you that example in the last lecture on plasma membrane. I also opened up this chapter giving you that example, okay? That means that neurotransmitter binds, it directly opens that channel, okay? However, there are other neurotransmitter receptors that are not ligand-gated ion channels, okay? One specific group of these are called G protein coupled receptors, okay? So that's a major group. And so they'll link cell surface receptors to intracellular responses, okay? So we'll talk about those types of receptors and the signaling of those, okay? It says peptide signaling molecules include peptide hormones, neuropeptides, and polypeptide growth factors, right? So these are other types of signaling molecules, okay? For instance, I'll give you it gives you some examples of peptide hormones there. Insulin, glucagon, pituitary gland hormones, growth hormone, FSH, follicle stimulating hormone, prolactin. These are all peptide hormones, okay? So they all bind to receptors that sit on the cell surface, okay? So these are examples of other signaling molecules. And so some examples of peptide hormones, okay? Also, if you think about neuropeptides, right? Enkephalin, endorphin, right? So if we think about, that's the endogenous opioid system. Those are endogenous peptides, right? Neuropeptides that are involved in perception of pain. And sometimes you'll see these same thing kind of considered as neurotransmitters, okay? But the point is that they're peptides, right? Small proteins, essentially, think of it that way, okay? So these are just some examples. Again, does anyone need to memorize these? No. Should we know that maybe endorphin, right, or enkephalin are neuropeptides? Yeah, maybe. But you don't have to worry about memorizing any of these, okay? But these are just giving you just a list of some examples, okay? And you can actually look here too to see the activities of some of these. Okay, so I just said this, right? It says neuropeptides are secreted by some neurons. So like I said, enkephalins, endorphins, you can consider them as neurotransmitters, okay? Because they act as neurotransmitters, but they also act as neurohormones. And I already mentioned this. They are involved in analgesia, right? Or in pain perception. They are in general inhibitory, right? So they sort of decrease the perception of pain, okay? It says they bind to the same receptors on brain cells as morphine, okay? So these are and again, you think about like, I'm sure we've heard the term endorphin before, like runner's high, right? That's due to the increase in endorphins that are released, okay? And again, they act as natural analgesics in that they decrease pain response, okay? All right, so again, we're giving you more examples here. So in terms of examples of peptides, right? Neuropeptides, okay, enkephalins and endorphins. If we talk about peptide hormones, here are some examples of peptide hormones: insulin, glucagon, okay? Now, some other examples, growth factors here, right? Nerve growth factor, okay? NGF, epidermal growth factor, EGF, okay? NGF, as the name implies, is a member of something called neurotrophins. And basically, these are all involved in regulating the development and survival of neurons. Epidermal growth factor, or EGF, in general stimulates cell proliferation. It says it's the prototype of the study of growth factor. So a lot of times, I mean, this might be some kind of factor you could add. Remember how we did cell culture, right? If we had our cells in culture and we added some EGF to it, to the media, it would greatly stimulate the proliferation of those cells. Okay? Oh, that's a good actually experiment maybe to do for next year. So instead of trying to kill our cells with ethanol, right? Maybe we try to promote their growth using a growth factor or something else that might promote their growth. Okay, another example of a growth factor, PDGF, platelet-derived growth factor, okay? So this is actually in blood platelets, and basically it's released during blood clotting at the site of the wound. And that will then stimulate proliferation of fibroblasts to basically contribute to that regrowth of damaged tissue and to basically form this sort of plug and induce the formation of a clot. All right, more examples here, cytokines. Okay, so again, these are peptides too, right? These are all examples of peptides, okay? Different types of peptides that act as signaling molecules, okay? So don't get lost in kind of the laundry list here. Cytokines, okay? So these regulate the development and the differentiation of blood cells and activities of lymphocytes during immune response. So I always think of cytokines as immune related molecules, okay? So you can think of them as immune system cells. So if there's some sort of injury or infection, right, immune system cells will release cytokines. And those cytokines then may be stimulatory to make more immune cells to be produced, or some of them may be inhibitory, okay? But they're in general, think of them as immune related molecules, okay? It says membrane anchored growth factors. So there are, remember how I showed you that sort of direct cell to cell signaling, okay? There are growth factors that are anchored in the membrane, okay? And so that's where you would see this direct signaling and the direct cell to cell interactions that we talked about in the beginning of the chapter, okay? All right. All of these types of signaling molecules, peptide hormones, neuropeptides, growth factors, can't cross the plasma membrane. Okay, they're peptides, right? They're small proteins. They're big molecules. They are not going to cross that plasma membrane. Okay, so they all bind to cell surface receptors. All right. Again, we've mentioned this about a million times. Maybe this is the fourth time. Abnormalities in growth factor signaling are the basis for many diseases, including many cancers. Okay, so you can, again, if you're wondering why the heck do I need to know about this stuff? Well, here's a reason why, right? And here's a reason why scientists might study the same pathway for their entire lifetime, because potentially one of those molecules in that pathway could be a target for cancer treatment, right? Could provide you with an explanation as to why or as to how a certain cancer is formed, right? Maybe it's due to one specific defect in a signaling, in a molecule that's part of a signaling pathway, okay? So again, try to kind of put things into perspective. You know, if we talk about drug discovery, you have to know every single component of a pathway to know how to target it, right? So if you don't study these pathways and know all of the components, how could you ever design a drug to target a specific component if you don't know them? Okay, so that's my little side note there. All right, it says most ligands responsible for cell-to-cell signaling bind to surface receptors on target cell. right, on target cells. I gave you some examples in the beginning of there are nuclear receptors, right? Steroid hormones can bind to nuclear receptors, and those then act as transcription factors and sort of directly alter gene expression. But for the most part, most molecules will bind to receptors that sit on the cell surface and then activate some intracellular signal transduction pathway, okay? So it says the surface receptors regulate intracellular enzymes, which then transmit signals from a receptor to a series of intracellular targets. All right, so like I said, we're going to look at some different varieties or different examples of these. Okay, so it says the targets of signaling pathways frequently include transcription factors, meaning all the way down the line, after a whole bunch of things were activated along the way, ultimately that may lead to the activation of a transcription factor, okay? But it is not direct like those nuclear receptors, okay? So let's look at some of these examples. So ligand binding to receptor initiates a chain of intracellular reactions. I think I said that a million times, which again would ultimately lead to reaching the nucleus and altering gene expression, okay? So this is down the line, okay? G protein coupled receptors, like I mentioned before, are the largest family of cell surface receptors. There are many neurotransmitters that bind to G protein coupled receptors or signal that way, okay? And so the reason why they're called G protein coupled receptors is that there are these guanine nucleotide binding proteins that are essentially associated with the receptor, and they're called G proteins, okay? So that's where the name comes from. All right, we're gonna look at a picture in a second. Binding of the ligand induces a conformational change that allows that cytosolic domain to activate the G protein, okay? It's gonna make more sense when we look at the picture. That activated G protein then can disassociate from the receptor and goes and carries its signal to some intracellular target, okay? So again, and I think I might have shown you this, there's an animation that kind of shows you this, okay? And now just remember, this is the beginning part. There are lots of things that are going to be happening downstream of this, okay? And we'll talk about that probably in a few slides. But here, your hormone, right? Hormone in the bloodstream binds to its receptor. This is a G protein coupled receptor. When the ligand binds, it basically causes this is the G protein, beta, gamma, and that's alpha, right? Subunit, okay? And basically what happens is the subunit will disassociate, those subunits will disassociate from each other, okay? And basically that alpha subunit will be activated. So that's the activated part, and it will go and will activate adenylyl cyclase, okay? Which will then cause an increase in cyclic AMP. What's happening here is a very sort of common sort of mechanism that occurs when a ligand will bind to a G protein coupled receptor. Okay, so there's more. Okay, so this basically describes what I just described here. Okay, but again, there's more that's going to happen downstream of this. Okay, all right, so again, 3 subunits, alpha, beta, and gamma, and that's why they're called heterotrimeric G proteins. Okay. Again, this alpha subunit binds guanines, which regulates that G protein activity. In the inactive state, the alpha is bound to GDP, okay? I guess it's not really showing you that. Bound to GDP, but once that ligand or that hormone binds to the receptor, it causes an exchange of GTP for GDP, and then that's how they dissociate, okay? So I believe... yeah, we can see that there then. Okay, bottom line is if you look here, right, which I guess is sort of left out here, is that this alpha subunit is associated with GDP, okay? Once the ligand binds, you have conversion of GDP to GTP, which essentially causes this conformational change and allows this alpha subunit to dissociate, okay? And then go have effects, other effects like activating this adenylyl cyclase, okay? All right. Okay, it says a large array of G proteins connect receptors to distinct targets. Okay, it says in addition to enzyme regulation, right, like that adenylyl cyclase that I just showed you here, okay, the activated G protein can also go and interact with ion channels. Okay, so it says example, action of neurotransmitter acetylcholine on heart muscle. Okay, so here's that example. So it says heart muscle cells have acetylcholine receptors that are G protein coupled. Okay, so again, we're thinking about a neuron stimulating a muscle, but except here we're talking about specifically heart muscle. Okay, so when that acetylcholine enters into the cleft, right? Remember I gave you the example of the acetylcholine binding directly to the ligand gated receptor. That was just hypothetical, okay? If we want to talk about exactly how acetylcholine signals here in heart muscle cells, this is the mechanism, okay? So the alpha subunit of that G protein, okay, this G sub I will inhibit adenylyl cyclase. So before we're showing this actually activating it, okay? In this case, it's going to inhibit it. The beta gamma subunits will actually open potassium channels in the plasma membrane, which slows heart muscle contraction, okay? Oh, I guess I didn't show you guys a picture of that. All right, I guess I'm just giving you another example. So I think I tried to pull out, or I tried to not have so many diagrams in here because it would get so confusing. So I just wanted you guys to have, you know, a few examples of signaling pathways to sort of focus on, okay? But I'm sort of mentioning some other types of examples within this, but not showing you the detailed picture, okay? The reason why I'm mentioning this is because if you notice here, again, this is showing you this alpha subunit going to activate the adenylyl cyclase. Well, in the case of acetylcholine binding to G protein coupled receptors in heart muscle cells, it's a different mechanism, right? So the alpha subunit actually is inhibitory, and it will go and inhibit adenylyl cyclase here, okay? So it would inhibit this instead of activating it. And then the beta gamma subunit will actually go and affect potassium channels and cause opening of potassium channels. So if you go back and think about what we talked about with action potentials, right? I don't know if I even want to go there, but if you open potassium channels, what does that do? Potassium goes out of the cell, right? That causes that membrane potential to become more negative, which essentially, it's being inhibitory, right? So it says it slows muscle contraction. So it's leading to that cell to not be activated, right? No, it's going to be less likely to generate an action potential and cause contraction. Okay, so if you wanted to try to piece some of these things together, but don't worry about that. I'm not expecting you guys at this point in time to understand that fully. Okay, the point of this is just to give you another example, right, or another situation that involves a G protein coupled receptor like this. Okay, however, once the ligand, which in our example case is not a hormone, it's acetylcholine. Once it binds, this alpha subunit will go and actually inhibit this. And then the beta and gamma subunit here will actually go and cause a ligand gate, or cause a potassium channel to open. Okay, so you can see that dependent on what type of receptor this is sitting here, what type of target cell it is, what type of ligand binds, it's going to have a different response inside the cell. Okay, so that's the point I'm trying to make. All right. Okay, now this example is an example that I've given you before. Okay, so glycogen metabolism. Okay, protein kinase A phosphorylates two enzymes. Phosphorylase kinase is activated and in turn activates glycogen phosphorylase, which catalyzes glycogen breakdown. Okay, we talked about this before. I've given you this example before. It says glycogen synthase is inactivated, so that you're not making more glycogen, because that's not what the cell wants to do. It wants to break down glycogen to have more glucose available. Okay, so again, this is the same example that I gave you before, but now you can look at this signaling pathway. So here's epinephrine, right? Binding to what? A muscle cell, right? Okay. And what happens there? So again, this is a G protein coupled receptor. Okay. You can see here this activated G protein here, this alpha subunit, will go, and in this case, it's going to activate adenylyl cyclase. And that leads to an increase in cyclic AMP, which then will go and bind to this other molecule here, this protein kinase A, which would essentially leads to activation of another kinase here, okay? Which then phosphorylates this glycogen phosphorylase, leading to the breakdown of glycogen, okay? So what we want to appreciate here is this series of events that's happening when this binds. Again, do you need to memorize this? No way. Do not memorize it, but we need to understand what's happening here and be able, again, to understand sort of each step and explain what's going on here, okay? Or be able to answer specific questions about this. You would have this picture, okay? I don't expect anyone to know exactly what's going to happen, you know, at each step here from memory, okay? All right. Yeah, so particularly here, if you can kind of see here, this C with the R and the C, I just wanted to just sort of double check what that is. So this is regulatory, this is catalytic. So once the cyclic AMP binds here, it essentially causes the catalytic subunits of this protein kinase A to be released. And then that's what actually leads to that phosphorylation, okay? Or leads to the activation of this phosphorylase kinase. All right, so again, an example, again, we want to understand, we don't need to memorize. Impossible to try to memorize all of these things. All right. Now, just a little bit more here in terms of kind of other things that can happen. So it says signal amplification. So it says binding of a hormone molecule leads to activation of many intracellular target enzymes, okay? Not just one. It says here, right, our example here, each molecule of epinephrine, right, that binds to a receptor, okay, it would activate one receptor, but each receptor may activate up to 100 molecules of G sub S, meaning this activated G protein here. So it's not just essentially one molecule and one sort of signaling cascade that's activated. Many are activated just by the binding of one ligand, okay? Okay, so that's what this is saying. It's amplified. The signal is amplified, okay? Binding of one hormone to one receptor, what's happening inside that cell after that point is amplified maybe about 100 times, all right? So that's what this is saying here. So synthesis of many molecules of cyclic AMP, not just one, okay? Each molecule of protein kinase A phosphorylates many molecules of phosphorylase kinase, okay? Not just one, many molecules that then can phosphorylate or activate many molecules of glycogen phosphorylase, okay? So one of the reasons why this is sort of set up biologically is because the binding of one ligand to one receptor can actually lead to a great amplification of response here. If it was just one single sort of switch on off, right, so say like it was just epinephrine binds to the receptor and then it just activates this. Well, you wouldn't have all these different points where regulation can occur. You also wouldn't have this amplification that we're talking about, okay? So that's one reason why things are set up that way. All right. Here is sort of another kind of a little bit more about cyclic AMP and some of these, signaling pathways or some of these molecules that are involved in a lot of these signaling pathways, okay? It says increases in cyclic AMP, like I said, that's something that's always kind of, if you think about a G protein coupled receptor, that's going to be stimulatory. This first part happens or is the same in a lot of instances, a lot of examples. But then downstream of it, the targets may be different, right? So this is saying, okay, when you increase cyclic AMP, right, there's going to be a whole bunch of other things that happen downstream, okay? So this is one example of something else that might happen. It says that increases in cyclic AMP will activate transcription of genes that have a regulatory sequence called CRE, or cyclic AMP response element. Because remember I said to you before that this is one example, but sometimes what's happening downstream of these pathways is that transcription factors will be activated, okay? So now we're sort of getting into that example. So it says the free catalytic subunit of a protein kinase goes of protein kinase A, okay? So like if we go back here, we had this protein kinase A, right? So again, think of it, again, you start can start the pathway the same in a lot of different examples, but downstream events are going to be different. So I'm sort of talking about an alternate pathway from here, okay? And it would be not from the same situation, but in an example where it's a different type of G protein coupled receptor, a different type of ligand that binds. Again, what happens up here might be the same, but what happens down here will be different, okay? So we go back to that, sorry. So the free catalytic subunit of protein kinase A will go to the nucleus and phosphorylate transcription factors, okay? Like CREB, right? And so phosphorylation of that CREB will lead to recruitment of co-activators and other types of, an expression of other types of genes, right? Okay, so it says, regulation of gene expression by cyclic AMP plays important roles in many aspects of cell behavior. So again, there are many situations where something like this would happen, okay? So again, it's just sort of giving you this example, right? Arbitrarily of some hormone binding to its receptor. Again, what's happening up here, I hope you all agree, looks the same as what's happening up here in this example of epinephrine, okay? When we're talking specifically about epinephrine, this is the pathway that it will follow, okay? That ultimately it's this glycogen phosphorylase that becomes activated, okay? Because again, that's the response that's makes sense biologically in terms of what needs to happen here, right? So we're talking about fight or flight. This is its effect on muscle cells. Well, you need to provide as much glucose as you can for those muscle cells to be able to work, okay? And so that's the ultimate response here. However, in other situations, there's going to be different responses that occur here. And again, we're talking about the same type of receptor being in that it's a G protein coupled receptor, but it's not an epinephrine receptor now, right? It's not epinephrine that we're talking about here. We're talking about other examples, all right? And again, you could talk about 8,000 different examples here. So we're sort of generalizing a little bit. But in many of those cases, what will end up happening downstream is not that a certain enzyme would be activated, but that a transcription factor will be activated and transcription will be affected, okay? But again, what I want you to notice is that the pathway sort of starts off the same way, but it's very different in terms of the way it ends up, okay? So again, you see adenylyl cyclase is increased, cyclic AMP, right? That leads to an increase in cyclic AMP. That cyclic AMP then essentially activates the protein kinase A. Remember, this catalytic subunit then can be released. And in this case, it's actually going to go into the nucleus where it's going to activate a transcription factor. Okay, and so this CREB is this transcription factor. This is also showing you a co-activator. Don't worry about that. The whole point is, or what I want you to understand, is that this is now going to activate a transcription factor. And we also should remember what a transcription factor is. It is a protein that binds to DNA that alters transcription. Okay? So here's another thing that cyclic AMP can do or can lead to. Again, think about this. It starts off the same way. Increase in cyclic AMP can do many different things. Here's another example. It can directly regulate ion channels. Okay? So it's a second messenger in sensing smells. And in fact, there is there's a little excerpt, and I'm not even going to bother putting it up because we got a lot going on right now. But it'd probably be something you might want to look at. But basically a key experiment that talks about G protein coupled receptors in odor detection. Okay. So odorant receptors are G protein coupled, and they stimulate adenylyl cyclase, which leads to increased cyclic AMP. Again, if you think about G protein coupled receptors in general, ones that are stimulatory, for the most part, they're going to increase cyclic AMP. And then what happens downstream of that is going to be very different depending on the situation. Okay, in this case, for these odorant receptors, the cyclic AMP will actually open sodium channels. Okay, and that will essentially trigger an action potential. Okay. All right, so are we all right with that? I gave you sort of some examples. I guess the take-home message here is to sort of understand Okay, this is sort of the beginning. If we talk about a G protein coupled receptor, what's happening here in general happens in most cases. Again, if we're talking about stimulatory G protein coupled receptors, okay? Once cyclic AMP is increased, though, what's going to happen downstream of that is going to vary significantly. And I gave a couple examples. We gave one, right? So let's skip over this for a second. We gave one where we're talking about epinephrine binding to muscle cells, and the ultimate goal or ultimate effect is the activation of this enzyme. And then I gave you another example where we see a transcription factor that's activated downstream of that increase in cyclic AMP. Also, remember that not all of these G protein coupled receptors will activate adenylyl cyclase and increase cyclic AMP, okay? There are some that are inhibitory here, okay? And so that's what this is telling you here, okay? Right? So this is why I gave you this example, was to just show you that sometimes that alpha subunit can be called something called a G sub I, which means it's inhibitory. And instead of activating adenylyl cyclase, it will inhibit it, okay? And leave it at that. And then again, what's going to happen downstream of that is going to, again, be very different than those other examples we gave, okay? So I would say I spent a lot of time talking about G protein coupled receptors. That probably means that they're important. So we should understand those examples that I gave about signaling through G protein coupled receptors and some of these downstream pathways that get activated as a result of a ligand binding to a G protein coupled receptor. Again, remember, there are thousands and thousands of different pathways that can become activated depending on what ligand you're talking about and what specific receptor, okay? So even for instance, epinephrine, right? Or really most neurotransmitters that you might think of, or hormones that you might think of, usually they don't just have one receptor type. There are multiple receptor types that will then initiate different responses too, okay? So it's very, very complex. All right, so let's move away from the G protein coupled receptors and just give you a few other examples of different types of receptors, okay? And again, this is really just meant to show you that there are different pathways that exist, and again, to appreciate how complex, you know, this really is, okay? But in terms of really understanding the mechanism and understanding the details, those G protein coupled receptor examples that we just talked about, you really should understand those, okay? For tyrosine kinases, okay, these are cell surface receptors that are directly linked to intracellular enzymes, okay? So like I said, these tyrosine kinases are sort of the largest family of these types of receptors, okay? So it says they phosphorylate their substrates, okay? So again, receptors, tyrosine kinases, okay, include receptors for most growth factors, okay? So I gave examples of sort of peptide growth factors, all right? Or when we were talking about peptide molecules, growth factors were a class or of example that we gave, right? And I mentioned EGF and NGF and PDGF. Well, these don't signal through G protein coupled receptors. Their receptors are tyrosine kinases, okay? All right, and so that's why we're going to talk about these. So it says the human genome encodes 58 receptor tyrosine kinases, okay? And there's probably more than that, maybe not. 58, there's probably different subtypes that basically include the receptors for, like I said, these growth factors that I mentioned before, EGF, NGF, PDGF, insulin, and other growth factors as well. Okay, so it says binding of the ligands, binding of those growth factors, say like EGF or NGF, to the tyrosine kinase receptor, okay? They bind to these extracellular domain, and then basically that activates these domains that face inside the cell, okay? And so it says it results in phosphorylation of both of the receptors, both the receptors and in intracellular target proteins, okay? So we're gonna look at a picture of this in a second, okay? So the first step that actually happens in sort of these pathways is a step called ligand-induced receptor dimerization. Again, here's dimer again, right? Here's dimerization again, meaning these two receptors are just going to come together to form a dimer, okay? And when that happens, it results in something called autophosphorylation, okay? So basically they phosphorylate each other here, right? So here's your growth factor, right? So here's your tyrosine kinase receptor sitting in the membrane, right? No ligand here, but once this growth factor comes along, it actually binds to these two receptors at once, okay? And so this is the dimerization part. So it's only a dimer when the ligand binds. Now, once these two receptors come close together with one another, they phosphorylate each other, okay? So that's sort of the first step in this pathway, signal transduction pathway, is this phosphorylation that occurs, okay? So we call them, remember, they're tyrosine kinase receptors. What are kinases? They are proteins that phosphorylate other proteins, okay? And so that's what these guys are doing. All right. So so this is called autophosphorylation, okay? And so it says it has two roles, okay? Phosphorylation of tyrosine in the catalytic domain increases protein kinase activity, okay? Also, this phosphorylation will create binding sites for other proteins, okay, that will transmit signals downstream of those receptors, okay? So it sort of preps, so it basically increases the protein kinase activity, but then it also makes it able to, essentially to bind other downstream effectors. Okay, so that was our example for growth factors, right? Or if we talk about growth factor receptors, okay, they are receptor tyrosine kinases. I'm not going through to show you essentially the downstream cascades that are activated, okay? Just showing you sort of the initial effects of or sort of how a growth factor binds, the fact that these receptors form a dimer and then they autophosphorylate each other. That then will lead to activation of a whole bunch of other proteins downstream of this, okay? And then we'll have effects on that target cell. But we're just not going to talk about all those specific details. All right. Okay. Another type of molecule, signaling molecule that I mentioned in the beginning of the lecture, right? Were cytokines. Okay, so we want to give an example of how cytokines signal or what their receptors look like. Okay. So it says the cytokine receptor superfamily includes receptors for most cytokines and also for some other polypeptide hormones, okay? There is a specific pathway called the JAK-STAT pathway, okay? And so the kinases that are associated with these cytokine receptors belong to this family, this Janus kinase family or JAK, okay? Key targets of these JAK kinases are STAT proteins, okay? And these STAT stands for signal transducers and activators of transcription, okay? The STAT proteins are transcription factors with SH2 domains, okay? All right. So to continue on a little bit with this JAK-STAT pathway, and again, this is something that's a pretty well-studied pathway. And again, if we think about cytokines, these are immune molecules that are released by immune cells. If you think about during times of injury or infection or something like that, immune cells will release cytokines. There are pro-inflammatory cytokines, which will then increase proliferation of more immune cells. But there are also anti-inflammatory cytokines as well that will decrease proliferation. But for the most part, these cytokines are there to affect the proliferation of these immune cells, okay? All right, so continuing on in terms of what's going to happen when a cytokine binds to its receptor, okay? And so the pathway that will be activated is something called this JAK-STAT pathway, okay? So a cytokine binding to its receptor will activate this JAK-STAT pathway. All right. It says STAT proteins are inactive in the cytosol until cytokine receptors are stimulated. Then those STAT proteins will bind to phosphotyrosine sites on the receptor and are phosphorylated by JAK, right? We said JAK-STAT proteins. Those STAT proteins that are then phosphorylated will dimerize and they will move into the nucleus. And think for a second, what do you think they're going to act as when they go into the nucleus? As transcription factors. Okay, and so this is giving you an example and showing you this JAK-STAT pathway. Looks different, very different than our G protein coupled receptors and what's happening downstream of them. Okay, so here cytokine binds. In the same way we talked about growth factors binding, very similar, right? You get this dimerization that occurs. Okay, you get basically phosphorylation that's going to occur here. These STAT proteins ultimately bind to this sort of phosphorylate, right? So the JAK and the STAT sort of work together where you have these STAT proteins sort of binding here once it's phosphorylated, and then they join together to form a dimer, right? That's what we said here. The phosphorylated STAT proteins then dimerize, okay? So JAK sort of, think of JAK as phosphorylating those STAT proteins, right? And then once they're phosphorylated, they then are able to form this dimer. That dimer then goes into the nucleus, and what does it do? It stimulates transcription, okay? If this JAK wasn't around or there was an issue with it and the STAT proteins weren't phosphorylated, the dimer would never be able to form. It would then never be able to bind to the DNA, right? It only can enter the nucleus in this state. It only can bind to the DNA when it's in this dimer state, okay? So again, this is a pretty well-studied pathway, this JAK-STAT pathway. This is showing you, I guess I probably should put this, a little out of order. But this is showing you what's going to happen downstream of a growth factor binding, right? So if we go back here, I guess the reason why is we were kind of talking about the beginning part of this receptor dimerization of these growth factors, which is similar to cytokines. I showed you what's happening downstream of those cytokines. Here now what's happening, right? Wait, I think I talked about, we talked about that SH. Where was that? I don't even know. All right, whatever. I thought I mentioned this. Yeah, here. This is showing you. This mentioned this SH2 domain here. So here, right? What this is showing you again is sort of what can happen downstream of this growth factor binding. Okay. Right, remember this was sort of this autophosphorylation that occurs. You have this SH2 domain that might bind here, and a whole bunch of other things are happening downstream. So you remember these, we talked about RAS proteins probably before. Essentially, these are proteins that are associated with GDP and GTP, right? And you can see once it gets converted into that GTP form, it's going to activate another protein, okay? Which will then, again, have downstream effects. Ultimately, here, once you get down to these are all different kinases, right? MEK, ERK, these are all different types of kinases. But ultimately, where's it going to go? It's going to then go into the nucleus and probably alter transcription, okay? Really, I don't want you guys to focus on this picture, to be honest. I just wanted to sort of show you it as an example of another type of sort of intracellular response that's going to happen. But again, you don't need to know details, okay? The other thing is it sort of sets up for this next slide. So I'm sort of sorry, it took me a second to realize why I put this in here. I don't want you guys to focus on details in this one, okay? But what I want you to appreciate, I guess, is that when a growth factor binds to its receptor, that intracellular cascade that's activated is different than this, right? This is particularly showing you this cytokine example, okay? And again, for different cytokines, right? Depending on what cytokine you're talking about, this is going to look different sometimes, okay? This is just one single example. Same thing here. Different growth factors, different growth factor receptors, different pathway. There's going to be similarities, but there's also going to be differences. All right. So this is just showing you here, if you kind of look at two different stimuli here, one being growth factor, right? The other one being a cytokine, all right? And kind of showing you some of these things that are activated. And we saw some of these a little bit when we were talking about the cytokines. We were focusing on that JAK-STAT pathway, but like I said, different cytokines, different cytokine receptor, different pathways that might be activated. I guess what I want you to focus on here, the reason why I put this figure in, try to sort of get your eyes off of all these different proteins and that are being activated kind of along this, right? Maybe kind of just look here in terms of what types of things are going on. So here's your stimulus, right? A growth factor or an inflammatory cytokine. When you're going to call it inflammatory or pro-inflammatory, it means it's going to lead to proliferation of more of these immune cells, right? And inflammation. Okay. You can see here that these are just different types of GTP binding proteins, right? So GTP binding proteins, right? Okay. This is right. So these are just all different examples, and there are many others as well. And then these are some other kinases that become activated as you sort of move down here. It's calling it upstream kinases because these are being considered as downstream kinases, right? So upstream versus downstream. Okay. So ERK being a downstream kinase. And again, if you look here, here's MEK, right? This is more upstream than ERK, which is more downstream. Okay, so when you think about upstream versus downstream, make sure you understand those terms. And then you can see what very different types of responses, right? Because that's dependent on what types of intermediate molecules are activated in these pathways. Okay, so here you see that when a growth factor binds to its receptor, it's going to stimulate proliferation, differentiation, cell survival. When an inflammatory cytokine binds to its receptor, it's going to lead to inflammation and cell death. Okay? So, and why does it lead to different things? It's based on what proteins are activated in this signaling cascade. All right? And so that's why I like this diagram, because looking at it side by side shows you sort of what's binding, and then what types of effectors are activated, and then ultimately what type of response is being generated. Okay. Okay, another type of pathway. Okay, we mentioned that JAK-STAT pathway. Here's another pathway here, PI3 kinase, AKT pathway. Okay, it says it's based on a second messenger derived from the membrane phospholipid PIP2. Okay. And so this is again giving you another example. So like I said, depending on what growth factor and what receptor is bound to, you're going to have a different effect inside the cell. Okay. So here's another type of pathway that can be activated by a growth factor binding. Okay. So you can see here this PI3 kinase, right? So this is this PIP2, PIP2 that I mentioned is involved in this pathway as well. But you can see very different, right? Very different than this here. This was a growth factor binding, right? But this is a different type of growth factor. This is a different type of receptor. And I'm not going to even mention or go into which one it is or what, because that doesn't even matter. The point is to understand that there are many different types of pathways that can be activated. Okay, so here's another example. And you can see here this mTOR that's in here, right? This AKT and this mTOR. That next journal article summary involves mTOR. I will put up a separate recording to introduce that paper for you guys, because there's a lot going on right now. So I'm not even going to detour that way. All right. But just to kind of make you aware. All right. So this mTOR, so this mTOR pathway, it says the mTOR pathway couples control of protein synthesis to availability of growth factors, nutrients, and energy. So think about that. The cell is not going to increase the amount of proteins it's making if there is not enough growth factors or nutrients available to it, right? So it has to have a way of sort of tying those two things together. And so that mTOR pathway is involved in sort of coupling those two things, okay? So it says here, this mTOR C1 complex is activated downstream of AKT. That's what this was showing you here, right? You can see this AKT as being involved in this pathway. And it says it regulates cell size by controlling protein synthesis. So this is actually kind of showing you another sort of, or more to this pathway, right? So where are we here? We're AKT here, right? We're showing mTOR C2 here. Okay, if we go here, right, and we also saw this PDK1 was in this other example, this mTORC2, right? It's here, binding to this AKT here. Okay, and so you see that here. Then this is sort of showing you now what can happen here, so that whenever you see something like this is inhibitory, right? That phosphorylation of this AKT will inhibit this. This is showing you if it's low energy, there's not a lot going around, it's going to essentially lead to this to be activated, but then again, this will be inhibited. And it's kind of showing you that this mTOR is sort of involved in this sort of decision as to whether there's enough growth factors and nutrients available outside the cell and whether that cell should increase translation. Okay, so you can kind of see that the mTOR is sort of the factor that's going to decide whether, based on this information that it's getting from up here, whether translation should be increased. Okay, and again, there's too much going on here for me to even walk through the different steps of what's going on. So, I mean, if I was going to ask you something about something really complex like this, it would be maybe a specific question about some step here, right? And again, nobody needs to memorize this. But I think the idea right now, or sort of the take-home message, just to understand that this mTOR pathway, or this mTOR protein particularly, is involved in coupling the control of protein synthesis to what's available on the outside of the cell, right? So it's basically sitting here taking in this information that's basically involving or basically telling it what the nutrient availability and growth factor availability is outside the cell based on what's binding here, and then basically relaying that information downstream here to determine whether translation should be increased. Okay. All right. It says here, mTORC1 also inhibits protein degradation by regulating autophagy. So think about it. If we're talking about, we talked about autophagy before, if we're thinking about a molecule or something that's going to be responsible for interpreting information sort of from the outside, whether there's enough nutrients available, whether there's enough growth factors available, it then has to have the ability to kind of talk to other proteins involved in autophagy, and also in other proteins involved in increasing translation. So depending, right, if there's lots of nutrients, lots of growth factors available, then you would basically, that mTOR would increase translation. If there is not enough nutrients or growth factors available on the outside of the cell, the mTOR would decrease translation and might increase autophagy, right? Because what happens in autophagy, the cell can break down its own contents in order to get some energy, right? To make up for that nutrient or growth factor deficit that's happening, okay? So it says here, when cells are starved of nutrients, that mTOR activity will decrease, and that would stimulate autophagy, okay? Okay, so kind of think about what I just sort of mentioned there. So again, this is saying, okay, if there's not a lot of nutrients available, right? It says low amino acids available, whatever. Not a lot of nutrients, not a lot of growth factors outside of the cell. It's showing you here this mTOR, right? Activity decreases, right? So here, saying its activity decreases. Okay, and basically you're gonna basically get this that you're gonna have the autophagy increased here, okay? And so you're letting this autophagy pathway go on, okay? So when cells are starved of nutrients, mTOR activity decreases. That will stimulate autophagy. So like this is showing you here, when this is decreased here, this stimulates this process. To be honest, that's a little misleading, the way that that's drawn. Let me just check something really quick, because I want to just make sure that one second, I just want to see something if this in this yeah. So in the new version of the textbook, there is a slightly different diagram here, but it's still kind of potentially basically showing you the same thing. And so I guess I kind of like this just because it's more general and sort of shows you the effect on autophagy. This is essentially kind of showing you the same thing here, and you can kind of see there can be an offshoot of autophagy here, right? So if this is stimulated and it's basically going to lead to an increase in stimulation, it's then going to be blocking autophagy, right? However, again, if we have this mTOR as being inhibited, it's going to allow this pathway to occur. Okay, so like I said, it's a little I don't know, it's a little misleading the way it's sort of written here. But let's think about this as low amino acids as causing this activity to decrease. Okay, and so if this activity is decreased, oh yeah, so think about that, right? Let me explain this further. Think about it this way. If mTOR is active, right? If its activities increase, it's active, right? It will stop this pathway, okay? Right? That's what it normally does. However, if this mTORC1 activity is decreased, then basically this stopping here is not going to occur, and this is going to be able to happen. And so you would see autophagy increased. So I'll say that again. Normally, when mTORC1 is active or its activity is increased, it will block this at this point, right? So basically it would inhibit autophagy if there was high activity. However, when its activity is low, this inhibition is not going to happen and autophagy will continue, okay? And so if you look here, this is basically showing you, again, when you have a lot of this going on, all right, it's going to lead to an increase in translation. Or when you have an increase in activity in this mTORC1, think of an increase in translation. When you have a decrease, okay, it essentially ends up leading to more autophagy to occur. But you can kind of think of this mTORC1 as being a sensor, right? A sensor to what's happening outside, a sensor to nutrient availability. Okay, and then the sort of relay between what's actually happening in terms of is translation increased or is autophagy increased is dependent on the activity of mTOR. Okay, if that sort of helps. And so, like I said, I gave you a paper related to it. As you might imagine, it's kind of complex, right? If you look at this, it's like, what the heck is going on? That's why I don't want you guys to focus on these details here. But this is the reality. This is what it is, right? I just want you to understand that mTOR is involved in sort of the decision of whether autophagy should be increased or whether more translation should be occurring, okay? And acts as sort of a sensor to what's happening to the outside of the cell, right? I think before, in previous chapters, we talked about autophagy in that it would be increased if there weren't a lot of nutrients available. Well, how does the cell know that, right? And what's the mechanism? Well, it involves this pathway here, particularly this one protein called mTOR, okay? And so that's sort of where this is more detail here in terms of how the cell can sense what's happening outside. Okay, it says multiple signaling pathways interact with one another to form signaling networks within the cell. So again, we've talked about all these different pathways. Well, they don't just act independent of one another, okay? So it says a full understanding of cell signaling must include development of network models that predict dynamic behavior of interconnected signaling pathways. They talk to each other. There's crosstalk between pathways. So I'll leave you at that. They don't act independently, all right? There's lots of crosstalk that occurs.